Hello, everybody. Thank you for joining us today. This is Melissa with Parkinson's Resources of Oregon, and so glad that everyone is with us today. Just a little background, we're a nonprofit that is based in Portland, and we serve all of Oregon and Southwest Washington. We do have employees in Eugene and Central Oregon. And our nonprofit provides information and support and education services to people whose lives have been touched by Parkinson's disease. We host these online presentations in an effort to make information on a variety of topics available through live interactive video webinars that are free to anyone to access from anywhere. I hope everyone is safe and well during this time. We here at Parkinson's Resources, we are working to reinvent our programs to meet the needs of people in Oregon and Southwest Washington. Right now we have some virtual sing-alongs that will start tomorrow, and mindfulness series that starts today and will happen on Friday as well. We also have tele support groups this month. If you are interested or would like any information, please feel free to email me at melissa@parkinsonsresources. We are also open to any suggestions or needs that you may have. So before I get started and introduce our presenter, I just wanted to go over a few details. Please feel free to submit questions throughout the program in your box provided in your browser on the right-hand side of your screen. We will save some time at the end. Actually, today we're going to spend about a majority of time on questions, so feel free to start submitting those as we go, and we'll respond to those at the end of our program. And we'll get through as many questions as possible, but Dr. Mata has left plenty of time for that. If you are having any technical difficulty, please feel free to submit those in the question box as well, and I will respond as soon as possible. So for the topic today, we're talking about Parkinson's disease and genetics, and Nacho, or Dr. Mata, leads the neurogenetic lab at the Genomic Medicine Institute at Cleveland Clinic. I'm going to hand it over to him and appreciate everyone being here today.
All right. Good afternoon, everybody, and thank you, Melissa, for the invitation. I wish this would be in person. I love Oregon, but we'll do the best we can with this crazy times that we're living in. Just a little bit of a background. So I'm a geneticist by training. I'm originally from Spain. That's why I have a strange name. I trained at the Mayo Clinic in Florida, and then I moved to Seattle. I was in Seattle for almost 13 years before I moved to the Cleveland Clinic in the fall of 2018. So I haven't been here for too long. But I'm very excited to be here with you all. I've been working on the genetics of Parkinson's disease for about 20 years now. I did my PhD on that, and I'll tell you a little bit of story if we have time at the end of how I started working on Parkinson's disease. But I'm very connected with the patient community, and I really enjoy giving this talk. So again, thank you so much for giving me the opportunity to talk today. So I'm gonna be talking about several topics today. I'm gonna tell you a little bit about what we know about the genetics of Parkinson's disease as 2020. So some of the things that we've been doing in my lab and in other labs in this respect. Then we're gonna talk about why research is important, and especially genetic research. And then I'll end up the talk telling you how you can help if you're interested and you think that what we're doing is worth while your time and effort. So we know about Parkinson's disease for a long time. In the 1990s, I think, is when people started wondering why people were getting Parkinson's disease, and for a long time. And this is a paper that was published in 1987. For a long time, he thought there was an interaction between aging and environment. So at any point, people thought that there was a genetic component. They thought that it was some kind of environmental factors that we couldn't understand, and then aging, because as we aged, our chances to get the disease are larger. So that's when we were in the 90s. nowadays we know that there's a lot of risk factors. We still don't completely understand why some people get it and why other people don't, but we do know for sure that there's some risk factors. I mentioned age. Age is probably the most important. You know, the older we get, there's no doubt that our risk increases. Males, unfortunately, we have a higher risk as well. It doesn't mean that it doesn't affect females, but it's usually around almost double the risk in males versus females. And then there are certainly some environmental factors that have been associated with Parkinson's disease. Heavy exposure to pesticides, heavy metals, and related to that, living in rural areas, for example, or drinking well water instead of drinking tap water from a city where maybe the water is treated. And lately, there's been a lot of talk about head trauma. And this is like heavy head trauma where people get concussions, where they lose conscience. Some of those can increase your chances of getting the disease. Doesn't mean that it causes, but it definitely increases your risk. Inflammation processes, so having lots of fever episodes, things like that, they seem to also increase your risk. And very, very recently, people have been talking about microbiome, which is really the bugs, the bacterias and the fungus and all the stuff that we have in our stomach that helps us digest all the food, and it really helps with a lot of different processes. People think that really has an effect not only in Parkinson's but also in cancer and some other diseases. So again, the field is still debating this, but I just put in here as much factors as we know, and then also exposure to certain viruses. And it'll be interesting to see what the influence of this COVID-19 will have in the incidence of Parkinson's disease in the next 10, 15 years. I think this is very interesting data that we're going to be gathering from now on. There's also some protective risk factors. So not everything increases your risk. There's things that also decreases your risk. Smoking, we're not sure exactly why. We don't know if it's the nicotine, but it definitely has some protective aspects, and it protects the dopaminergic neurons for some reason, and caffeine too. With this said, I don't want to encourage anybody to start smoking. This is people that didn't smoke before the diagnosis. Once you're diagnosed, it doesn't seem to help that much, and also you can get lung cancer. So I don't, Parkinson's and lung cancer. I don't know. I think I wouldn't risk it. There's other things like exercise, for example, that definitely not only seems to be protective but also seems to help a lot with the symptoms. So I would encourage people to exercise more. And then taking some anti-inflammatories, again related to this inflammation processes that I said is an increased risk, seem to be also protective. And then the genetics, at the beginning, again, people didn't believe that it was genetics. I'm going to show you today that now we know that there's a lot of genetic component, but a lot of it is very small effect. So it's not Parkinson's disease is not a genetic disease like Huntington's disease or some other diseases where it runs in every single family. So everybody has a family member with Parkinson's disease. It increases the risk for everybody else in the family. And then one thing that I'm really interested in is how ethnicity actually plays a role. Because I study underrepresented populations, specifically Latinos. We want to know if Latinos get more or less Parkinson's, and if so, what are the reasons? Is it genetic or if it's something else? So overall, Parkinson's disease, as you probably already know, because if you have the disease or somebody that have the disease, that it's a very complex disease. It's known by its motor symptoms, but there's a lot more going on. And this complexity clinically is probably related to this complexity of etiology, of the cause of the disease. So I like to think about Parkinson's or genetics really in general as a plane cockpit. There's all these genes, all these buttons that do different things, and we know that they have different influences and the risk for different diseases, and it is kind of hard. We know most of the genes already, but we don't really know what they do. So basically, it's like if you walked into a cockpit, you see all these buttons, and you can see them all, but you don't really know. And the genes don't have labels. Like some of the buttons do. So you don't really know what they do. So what we're trying to do now in the genetics field is try to figure out what all these genes do. And not only the genes, but also the variants, the genetic changes that occur in each of these genes could have different effects. So for example, some of the effects you can imagine on a plane, like the air conditioning one goes off. It's pretty bad, but you can survive, right? It's not like the plane is gonna fall or anything. So that will be a risk factor, a genetic risk factor. Is a change in one of the genes that what causes is that it makes the system, so the cells don't work very efficiently, so they don't work as well as they could, but they're still working. So that means that you don't really go and have a disease straight away, but it definitely increases your risk. You can imagine that in this situation where there's no AC, but now imagine that you combine that with some environmental or other genetic factors, like there's no water on the plane and there's a flight that goes from LA to New Zealand, so it's a 13-hour flight. Now this change becomes a lot more problematic, right? Because now this small thing makes people can get dehydrated, so they can then get really sick just because of this lack of air conditioning. So there's genetic risk factors that can accumulate either between them, or there could be a genetic risk factor on top of the environmental risk factor, on top of the genetic risk factor, that can make this genetic risk factor even worse. So that's how genetic risk factors work. So they increase your risk to a level that then certain people will develop the disease and some people don't. But there's definitely some changes, and these occur also in Parkinson's disease, where the change is bad enough that the plane is going to go down really fast. So these are usually the changes that we observe in families. So they're changes that definitely have a very big impact on how the cell is functioning. In this case, the dopaminergic cells that are the ones that produce dopamine, and makes the cell not work properly, and then it started to create these problems that end up being Parkinson's disease. And the same thing is true for Alzheimer's and a lot of the neurological disorders. They're very complex, and in all these diseases, they're both kind of changes. There's changes that are very bad. Fortunately for us, those are the less frequent ones. But there's also small changes that combine between them, or with some environmental factors. Like imagine you have some genetic factors on top of pesticide exposure, or you work in a mine with heavy metals. So those two combined, then it can cause Parkinson's disease. So that's how you can kind of define how the genetics work. And this can also be seen if we classify patients between familial. So those are patients where there's at least 2 affected individuals in the family, and then what we call sporadic, which means that you ask the patient, and the patient says, I don't think I have anybody else in my family that has Parkinson's disease. And those are the most common. So about 80% of the patients don't report somebody else in their family affected, but there are certain, about 15-20%, where there are several people affected, and even within those, only a small subset of them have one of these really bad variants, like the plane is going to go down, which means that a lot of the descendants from that person have a really high risk, or a very likely likelihood of them developing the disease. So the good news is that very few patients will pass the disease to their descendants, but it does happen. And studying those families actually really helps us identify these genes, these buttons that you are like, well, we don't need to press this button, because we know what happens when we press this button, or we try to fix the button. So these families are very important to understand the disease, and to be able to create new therapies for Parkinson's disease. So I'll talk just a few. There's many genes that have been discovered. I'll talk a few about you probably heard about. So the first one is synuclein. Synuclein is probably the most important, but it's also one of the less frequent genes that are mutated. So it's one of the genes that have less genetic variants that actually cause the disease. A lot of them increase the risk if you have them, but they don't cause the disease on themselves. And this is the first gene that was discovered early in the 1990s. Late on the 1990s, almost in the 2000s. And patients that have this have very interesting forms of Parkinson's disease, where the age of onset is usually around 50, so a little bit younger than your normal patient, but also have very rapid progression and complication with cognitive impairment, and most of them will develop dementia at some point. Similar what an Alzheimer patient will be. We know that there's large families with this. There are not many in the world. It might be, I don't know, 20 families that have one of these. On top of the genetic variants can be divided in two. There are single nucleotide changes, but there's also when the gene is completely intact, there's no a genetic variant that might cause the disease, but you have several copies of the gene. That means that you're producing more synuclein than you should. And that seems to also be pathogenic. So it also seems to be a link to developing Parkinson's disease. And a lot of the families that we see have those kind of situations where they don't have a change in the gene, but what they have is that they have multiple changes. So they have multiple buttons for the same thing, and that causes a lot of problems. And the more copies you have of the gene, the worse the symptoms are. So you can see, for example, if you have, normally we have two copies, one that comes from the mom, one comes from the dad. So if you have one extra copy, so if you have 3 copies in total, your form of Parkinson's disease is kind of normal, and age of onset around 60s, cognitive impairment, but age of onset is very similar. If you have 4 copies, so if you have 2 extra copies of the gene, then your age of onset goes down from 60 to like 30. So the progression of the disease is much faster the more copies you have. So again, it's not very common, but it is the most important, and functionally is the most important because, as you probably all know, synuclein is the protein that is found on the Lewy bodies. So this protein aggregates that almost all patients with Parkinson's disease have in their brains, in their especially in the substantia nigra, where the dopaminergic neurons are. So these Lewy bodies appear. The biggest component of this is synuclein. So we know that synuclein plays a very important role in Parkinson's disease. The second gene is Parkin, and Parkin is a very, very specific gene that affects early onset. And when we're talking about early onset, it's like people that get the disease before 40. I forgot to say that synuclein is actually a dominant gene. So what that means is that you only need to inherit one bad copy, either from your mom or from your dad, and that will cause you to develop Parkinson's disease. In this case, Parkin is recessive, so that means that you have to get a bad copy for both your mom and your dad. So that means that in the family, sometimes it skips generations. So you don't see individuals that are affected in every single generation. But people that have mutations in this gene actually have very early onset. And the youngest I've seen is a 12-year-old with Parkinson's disease that had a mutation in one of these genes. So about 50% of patients that have early onset forms will have a variant in this gene. So it's also a very important gene. And you can see here in the table, there's more than 300 variants that have been identified. But again, most of them affect only one family. So that means that they're quite rare. Although overall, they affect a lot of early onset patients. So one conclusion from Parkin is to say that the earlier your age of onset, the more likely it is that you either have a very strong risk factor, either genetic or environmental. So a lot of people that have very early onset is because they work in mines, or they did something, or they were exposed to a lot of pesticides, or they do have a genetic variant. So I always tell people that if you're interested in doing genetic studies, the earlier your onset is, the more chances it is that you might have something in there. Then the third gene that is very important, and I think is probably in my mind the most important, and I was lucky enough to be working on this gene for a long time, and I was one of the people that worked on it at the beginning, is the LRRK2 or LRRK2 or dardarin, it's also called. And this is a very important gene. It's very long. It's also dominant, so you only need to inherit one RON variant, either from your mom or from your dad. There's a few variants in this gene. You can see some of them here, but as far as we know, most of the variants, we are not completely sure that they cause Parkinson's. Some of them increase the risk. Some of them are even population specific, so only affect Asians, for example, or they only appear in Asians. It's not that they affect in Asians, but it's they're variants that are only in Asians. But the most important variant for sure is this G2019S, which you probably heard about. So in the US, about 2% of the patients with Parkinson's disease will carry this genetic variant, and there's clinical trials ongoing for people that have mutations in this gene. It's a very important gene. It's found in almost every single country that has been studied. You can see here that only Korea and Taiwan are the only ones that don't have no patients with this mutation. Other than that, every single other country has carriers. And I'm not going to get into much detail, but we actually did a very interesting study a few years back, and we were able to track this mutation to one single ancestor. So most people that carry this variant actually come from one single person that carried this variant about 250 years BC, and then it got spread mostly during the Jewish diaspora everywhere else in the world. So it is a very interesting variant, and it helped us study migrations, for example. So it was very exciting to be involved in this study. The only problem with this variant, and the more people we genetically tested for this variant, we started realizing that this was going to be a problem, is that there's people that have the variant and still survive the disease. So in genetics, we say that this variant is not fully penetrant. So what that means is that there are people that carry the variant and still, skip the symptoms, and they might pass away from something else without ever develop Parkinson's disease. The number of those people is not high, but several studies, especially this one by Healy et al. In 2008, show that about when you're in your 80s, if you have the variant, you have about 74% chances that you will get the disease. So it's considerably. I guess the people that don't develop the disease, which is over 25% at the age of 80, is quite large. But then the older you get, these percentages start going up all the way to almost 100%. So it's a matter of time. More than the effect, I think, is a matter of having enough time to develop the disease. Some people have an age of onset that is even in the same family, that the age of onset could be very different. And this is telling us that there are probably other factors that are they're modifying the effect of these variants. One thing that happened around 2012, 2013 is that the price of the genome sequencing, so be able to read every single letter on your genome, went down so much that now it's very easy to sequence patients, the whole genome. So it's easier to find new genes, new variants, and this spiked a lot of studies in Parkinson's disease and other diseases. In Parkinson's disease specifically, around 2011, and it was almost like there was a new gene every couple of months or so. In Parkinson's disease. Again, some of them, or most of these ones, are kind of rare, so there will be 1, 2, 3, 4 families in the whole world with it. But again, the important part is that this is telling us what are the key components, what are the key proteins, the key buttons in our plane that really are making people develop Parkinson's disease. So they're very interesting and very important from the understanding point of view of the etiology of the disease, so we can develop new therapies. And I put a few examples of genes here. but the list is going up and up, and in fact we have close to 23, I think, maybe even more now. I think this is from 2018, so there's probably a few more now, a few more genes and these ones are the ones that are involved in the familial forms. So most of these, all these genes, variants in these genes will cause familial forms. So several people in the family will be affected by the disease if they have this variant. And you can see here that the onset could be early or late depending on the gene, and then some of them are dominant, as I said, so you only need one copy. Some of them are recessive, so that means that you need to have 2 copies. So that means that the chances of your descendants getting the disease because of these variants decreases every time a new generation comes. So there's a big list. But as I said, the familial forms in Parkinson's disease are only about 20%. So there's about 80% of you, of patients that do not carry one of these variants, but they do carry other variants that might have increased the risk for developing the disease. And these are the ones that I call genetic, or we call genetic risk factors. And again, these are variants that have very small effect, like the air conditioning going off on your plane. The plane is not going to go down, but several of these together with some environmental factors might actually be the cause of your form of Parkinson's disease. The interesting thing is that some of these genes are in the same genes that also cause the familial form. So synuclein, LRRK2, those genes actually have variants as well. And these variants typically are common in the normal population. So everybody's walking with a few of these, in their genomes. So they're more common, but the effect is small. But when you combine several of them, then that's when you get the disease. So the most important genetic risk factor for sure, and this is the other gene together with LRRK2, that if you only get 2 names out of this whole spills that I'm giving you, remember LRRK2 and also GBA, because there's also clinical trials for GBA, and in my mind is definitely a genetic risk factor that we need to keep in mind. It's rather common. It's about 5 to 10% of the patients with Parkinson's disease will have a variant in this gene. The risk is about 6-fold. So if you carry one of these variants, depending on your ethnicity, your risk goes from like 6 to 14 times higher. And also the patients that carry these variants have a very specific type of Parkinson's disease, very similar to the synuclein ones, where there's a lot of cognitive impairment, and it's linked to high diagnosis of dementia of all these patients, and it's usually quite fast as well. So even from the clinical standpoint, it's very important to know if you have one of these variants, so you know what your prognosis might be. So it's very important. And this gene, the association with Parkinson's disease, because actually this gene causes a different disease called Gaucher in a recessive form. So if you get 2 variants of this gene, you develop this lysosomal storage metabolism disorder called Gaucher disease. But if you carry only one of them, that's where your risk for Parkinson's disease increase. So in a lot of the families with Gaucher, we also see an increased number of patients with Parkinson's disease. So very interesting gene. But now we have the technology to be able to identify genetic risk variants all over the genome. So this is the last study that was published last year. You can see how many patients they actually use. Almost 40,000 patients participated in this study, about 1.4 million healthy controls. They looked at about 8 million variants in the whole genome, and so far is the largest study in Parkinson's disease to date, and they were able to identify at least 90 different variants that explain about 16% of the heritability risk for Parkinson's disease. So we're getting to the point where we know a lot about the genetic risk factors for Parkinson's disease. You can see here that some of the highest ones, so I'm not going to explain this Manhattan plot because it's kind of complicated, but basically the higher the dot is, the more the larger is the association with Parkinson's disease. So you can see the genes that we already recognize, like synuclein is here, GBA is here, LRRK2 is here. So some of the very important genes are already in here, and then there's a lot of a lot of a plethora of new genes that a lot of them we don't even know what they do, that we need to now follow up on, that seem to somehow play a risk on Parkinson's disease. And if you imagine a lot of these being buttons in your airplane, if you shut off a few of these, maybe one won't take the plane down, but if you knock enough of these buttons down, the chances of getting Parkinson's or the chances of taking the plane down, increase. So that's how you have to imagine risk factors, something that kind of accumulates and then increases your risk for the disease. So yes, as a summary of this introduction of the genetics of Parkinson's disease, hopefully I didn't go too fast, and if there are any questions, we'll answer them at the end. But in terms of trying to summarize everything, so the genetics of Parkinson's disease is very complex. There's a lot of genes that are involved, but all the genes and all the variants can be divided really in 3 different groups. And here we have the penetrance. So this is the chances of, if you have a variant in that gene, that you will get Parkinson's. So the higher it is, the higher the penetrance is, so the higher likelihood that you're going to develop the disease if you have a variant in that gene. And then here on the x-axis, we have the variant frequency. So this means that these are variants that are more frequent in the normal population. So we have 3 groups. We have our families, so patients with Parkinson's disease where there's a lot of people in the family, or not a lot, but more than 2 affected individuals. And then we have several genes. The most important ones are probably synuclein, Parkin, pink one, which I didn't touch on, but there was somebody asking, so I'll talk a little bit about it after the talk. LERK2. So some of these genes are included in this, where the variants are very rare, so not a lot of people carry them. But if you do carry them, you have a high likelihood to develop the disease. Then a lot of the genes, and here it will be the 90 that I was telling you about, where the frequency of the variants is not high, but I mean maybe 10, 15% of the normal population, of the healthy population, carry these variants. But they have very low effect. So yes, carrying 1 or 2 of these is not going to push you over the limit to develop the disease. But if you carry enough of these, or if you mix it with environmental factors, then that's when the disease shows up. And then we have some variants or some genes like GBA, or some variants in LER2 where they're not rare, but they're not really frequent either, and they don't cause disease all the time, but they cause disease enough that they're separated from this genetic risk factors. So those are very important to consider for sure. And if we put all this in a biological pathway, all these genes is telling us some of the things that are going wrong in the cells. So the protein cleanup, like the autophagy, which is the mechanism that the cells use to get rid of proteins that they don't want. The ubiquitin proteasome system, which does a very similar thing of autophagy. It just marks those proteins that need to be degraded. And then mitochondria is very important, and this is where PINK1, Parkin play a very important role. So this is the organelle that produces all the energy on your cell. If there's something wrong, then you don't have enough energy for the cells to function correctly. So with all these, we're putting little pieces of the puzzle to try to understand biologically what is going on in our brains and why some people develop Parkinson's disease. So hopefully that gave you a good intro on the genetics. A lot of people ask, so do I need to get tested? Is it worth for me to get genetic tested? So I can tell you that, for diagnosis, we don't really recommend. So people that don't have Parkinson's disease and they want to know if they have or if they are high risk, we don't recommend doing it because we don't know enough yet. There are very few cases where we are going to be able to affirm for sure that you either not going to get it or you're going to get it. There's so many variables that for diagnosis, we don't use it. We don't usually do it. We usually do it for people that are already diagnosed. We can use it to detect a possible mutation that segregates in the family. So if you have several affected individuals in your family, you might be a good candidate for genetic testing, just so you can find out if there's a reason for that. It could be also environmental. Remember that families also share the environment in many cases, right? So if there's something environmental there, that could be the reason. But to me, the last three are the most important reasons why somebody might want to get tested. One is because I was telling you about GBA, so prognosis. The more we know about some of these genetic variants, we know that some of them have a different prognosis. So for the clinician and the patient to know if they carry one of those variants and know, okay, well, maybe I will develop dementia in 10 years, so I might want to, start, exercising more and doing other things to try to prevent that from happening. Also, eligibility for research and clinical trials. I think we're now going to a field, and this is not only Parkinson's disease, but this is in every single disease, that patients are biologically very different. The causes are very different. So the clinical trials are going to be more geared towards people that carry a certain variant. And we're seeing this in LRRK2 and GBA, where there's now a specific clinical trials for patients that have this type of variants. So now knowing that you're a carrier empowers you to decide if you want to participate in some of those clinical trials. If you don't know if you carry, then you can't really decide if you're gonna be a good candidate for it or not. And then the precision or personalized medicine, which is a field that is, rapidly developing, which is telling us that we should be tailoring the treatments based on genetics to make it as efficient as possible. And I'll tell you just one slide about this. Before, just to tell you, as I mentioned before, that there are several clinical trials ongoing. There's at least two. This Denali one, I think, is in phase 2 now for LRRK2 carriers, and then GBA as well. Clinicaltrials.gov is a very good website to check the clinical trials. Also, your local or national foundation, Parkinson's Foundation, the American Parkinson's Disease Association, those are very good resources for information about these clinical trials as well. Or ask your clinician and see if they know of any local efforts to do clinical trials. And then one of the things that to me is the most exciting one is be able to use all this genetic information to take a group of people that don't have the disease and be able to classify them into different risk groups. So you can see that if you mix, and this can only, it doesn't have to be only genetics. You can also put environmental factors in there. And then if we're able to classify people into these different risks, we can try to attack the disease before it happens. One of the big problems with Parkinson's disease is that the person that develops Parkinson's disease, really the disease is going on for 20, sometimes even 30 years before you start developing any of the symptoms. So by the time you go to the clinic because you have a concern, it's too late to be able to prevent the disease. The damage is already done. So it's very hard to come back from that. So we're trying to find people at risk or people that are developing already the disease without any symptoms to be able to see if we can protect them so they never develop the disease. This is something that is definitely in everybody's radar, and I think the genetics can really help to be able to classify people into these different risks, and then you can approach people in a very high risk with some protective therapies that then you can do clinical trials on and see if any of those will help. And I think that's the future. And fortunately for us, because all these studies that we're doing in the genetics and the one that I told you about, the 90 variants, we're getting to the point where we can predict quite well if we take a group of people who might develop Parkinson's disease with a rate of about 60, 60, I can't remember what the number was. I think it's like 60 some percent. We can actually be, accurate in terms of being able to predict who's going to develop the disease or not. And there is an increased risk for people that are in high risk, almost 6 times more into people that are in high risk versus low risk. So we're getting there. We think that if we can get to 100,000 participants in our genetic projects, we can probably identify most of the genes involved in Parkinson's disease, and these models will be a lot more accurate, and then we'll get to the point of being able to develop new therapies. So how can you help? So this is the problem. The problem is that, you know, only 1% of the people in the United States with Parkinson's disease participate in research. So we need more. We need more patients to be able to participate in research, to be able to get the numbers that we need to be able to understand. And then a problem that it really concerns me is the representation of minorities, because all this information that I told you about, it's all done in European. So we don't really know if Latinos, if Asians, if Africans, if they have the same risk factors or not. And the studies that we've done in our lab with Latinos show that it looks like the risk factors might be actually different. So we definitely need representation of more patients, but also more minorities being able to participate in research. Another thing that people are working on is the average time for a new drug. So when we find a new gene, a new target for a new drug, we need the system to be more efficient to be able to develop drugs faster. And then one thing that I think is really easy to fix is that, you know, a lot of the healthcare providers and the patients don't really know if there's any research studies done in their area. So we need to advertise more. We need to make sure that all the information is in one place or in very few places where it's very easy for the patient to just go there and see, okay, and then look at all the opportunities that there are, and then, you know, decide if any of them fit your criteria, or if you know, there's some that are more involved than others. So you can, depending on your time commitment and whatnot, then you can decide if some of them are more interesting to you. Some people want to do the exercise trials, for example, because they're very big into exercising. Or some people say, look, just take whatever you need from me, you know, a CSF, blood, whatever, you know. And then some people just want to do surveys at home, which are also very helpful to try to find what is going on in terms of symptoms and your habits, to be able to learn what of our normal habits might be increasing or decreasing our risk, or improve our symptoms, for example. So the solution is to participate, be active. There's, you know, nothing of what I presented today could have been possible without the participation of you guys, of the patients. The patients are, in my mind, the key to find the cure. There's no other way to do this. And I'm just gonna present just a couple. I'm not biased. I don't get paid by any of these institutions to show what they're doing. But I am involved in this Parkinson's disease, Parkinson's Foundation initiative, which is called PD Generation, and it's all based on genetics. And what they want is they want patients to be able to access a system where they can get tested for the most important genes. So only out of all the ones that I told you, they only test for the 7 most important genes. But the idea is that they can give the information back to the patient, so they empowers them to be able to decide if there's a clinical trial that is targeted to one of those genes to be able to participate. The preliminary study just finished up, and now they're going to start opening more. You saw that there was only a few centers that were doing it. Now they're increasing this to more centers all across the U.S. To be able to do this, and there'll be genetic counseling involved. So it's a very cool project. So I highly recommend to have a look at this. And my role on this is to try to engage the minority communities to be able to participate in this, because again, we don't want, you know, all people to participate in this project to be from one ethnicity, which will give us some information, but it won't give us information for everybody, and we really need information for other populations. So you can check it out. You'll get the slides after the talk, so the links will be in there. The Michael J. Fox Foundation has a couple of projects as well. The PPMI project, which is heavily involved, but it's a longitudinal study, which is very, very important in my opinion, and is hopefully it'll generate a lot of good data. And then they also have the Fox Insight, which is a service that you can do at home. Then the NIH has also the PDBP, the Parkinson's disease biomarkers project, which tries to identify biomarkers of both disease and progression. And then here's the clinicaltrials.gov website that I told you about. that is a very good place to go find clinical trials that might be local to you. And then just to tell you, so because I was in Seattle, I was involved in one of the 10 UDOL centers that are all across the U.S. And I was participating with people at OHSU, which is a great group of movement disorder specialists, gay people. I mean, they have really a good group that does a lot of research, and this is still ongoing. So I put the links in there. I highly recommend having a look. That's locally a very good initiative to participate in. With that, if you guys want, I can either tell you the story of how I got started on Parkinson's disease, or we can start answering some questions. So it depends which way you want to go. Are there a lot of questions already? All right. So let me first. I'm going to answer a question that was sent before the talk. One was talking about PING1. So PING1, I didn't comment on it because it's much rarer than synuclein or Parkin, but it's also a recessive gene. It's very heavily involved biologically with Parkin. It usually also affects early onset forms, but less than 1% of the of patients have a mutation in this gene, so that's why I didn't touch on it. And then I think somebody also asked if there were, if I have Parkinson's, what are the chances of my my kids to have the disease? And hopefully I kind of answered the question before or not during my talk, but it really depends on if there are other family members, what is your age of onset. But in most cases, hopefully it got clear that in most cases it doesn't really pass from one person to another. There's a lot of other factors that are involved. But like any other disease, if I have some kind of family history, so my mom, for example, passed away from cancer, so I try to avoid things that are high risk. I don't know if genetically I'm predisposed or not, but I try to avoid. So that's what I usually tell people. Like, for example, if you have a family member with Parkinson's disease and you know that exposure to pesticides can increase your chances, don't try not to be exposed or protect yourself. Be healthy, although everybody should be healthy, right? But if you have a family history of one of these diseases, I would definitely take it a little more serious. But other than that, I don't think the risk is very high in most cases. Okay, so let's see. So is there a genetic connection between PD and Lewy body? If so, what are the chances my daughters will be carriers of their children or themselves? So there is a connection, and actually GBA is one of the genes that connects PD with Lewy body. So so Lewy body, I'm guessing they're talking about DLB or Lewy body dementia. So GBA is frequency of GBA is actually even more increased in patients with dementia with Lewy bodies. So we think that GBA is a gene that is a link between those two processes. So there is definitely, it's like we call it as an umbrella. So there is synucleinopathies, so diseases that develop accumulation of synuclein in the brain. We think that some of these genes might actually be playing a role in all of those. So there's definitely a connection, and the chance of the daughters to be carriers, I think I already answered the question. The chances are usually very rare unless there's a lot of affected individuals in the family. I would not worry too much. All right, so those are the questions. So I'll tell you then a little bit about the story, and to me this also shows the power of of the patient. So I started working on Parkinson's disease in a very small, well, not very small. It's about 300,000 people in northern Spain. My lab in Spain had two different research avenues. One was cardiovascular disease, which is the #1 cause of death because our diet is not the Mediterranean diet, so people there don't eat very healthy. So cardiovascular disease is a big problem. And then they have another area where there was only a couple of people doing research that involve neurological disorders, including Parkinson's and Alzheimer's. So they offered me to be involved in one of those two projects. I always found the brain incredibly interesting from my college years. So I decided, and I always cheer for the weaker. So I thought I would go for the place where there was not a lot of people already doing research on. So I started doing research on Parkinson's disease. Back then, this is the 2000s. The genetics of the disease were, most people were very skeptic that there was even a genetic factor for the disease. So there was not a lot of funding money for this. But my lab was very interested in studying this. So I started working on this, and I worked maybe for 8 or 9 months just for free. I just wanted to learn. I wanted to advance the field. So I was working for free. I was living at home. So I didn't really need the salary to survive. So I was fine working for free. And then there was a Parkinson support group, and you can see some of the patients here. This was the president. Well, this was the president at the time, and then Amada was the next president. And they came to us and they said, we want to help with Parkinson's research. So what can we do? And I said, well, we need two things. We need patients to participate in our projects. So if you can tell all your members to participate, that would be really good. And then the second thing is, if you can help us to find funding to do more research, that will be great. So they got into work. They said that convincing patients to participate was not a problem. So we managed to build a really large biobank of patients with Parkinson's disease samples. But funding was really, really hard to get. Nobody was banking on us funding anything interesting. So the patients themselves decided that they were going to fund a scientist to be able to do the work. So as part of the membership funds that they have, so members pay a fee to become members and take advantage of all the activities that they do. So they put part of that money towards funding somebody to do research. In that case, me. I was the first fellow for this fellowship that they created. There was a bank that then supported them as well and helped them with matching that effort. So that allowed me to really start working on research. And then I also felt that I was working for them. So I felt, that I owed them to go very frequently, talk to them, tell them the things that we were doing, but also the things that other people were doing. So they were, they were very excited about all the research that we were doing. These two pictures down here are pictures of the patients that came to my thesis defense, which, by the way, got the highest award in my university. And there was about 20 of them that came to my thesis defense to support our efforts. The newspapers. This is a news that came out on a local newspaper, and that was picked up by national newspapers as well. This was the first time that Parkinson's, well, actually any disease support group was funding their own researcher. After that, a lot of them copied a model. They still have fellows, so I think they're on their 6th fellow now in this last 20 years or so. 3 of them we are here in the U.S. Working, so we've been very successful thanks to their effort to be able to fund our research. So this doesn't mean that you guys need to start raising money to do research, but I'm telling you how, if you want to develop this, you have to take matters in your own hands, and if it's participating or what you're doing right now, which is increasing awareness. We're now in the Parkinson's Disease Awareness Month, right? So we need to make sure that people know that it's a very frequent disease, that there are needs that need to be met. There's funding agencies need to increase their funding so we can find a cure for this disease. So I feel really grateful to the patients, and that's why one of the reasons why I love to give this type of talks to patients and also caregivers of the patients. I never say no to those, and I still go back home and go see the patients, and they're very thankful. They gave me a golden pin last time I was there as one of the successful fellows from all their efforts, and it's been really a really exciting story. And I had an opportunity to change diseases a couple of times, but I feel like I need to do this for them and for all the rest of the patients that participate in our studies, and put a lot of effort into us being able to do this type of studies. So that's my little story that I like to tell people. Most times people feel very empowered about being able to do something like this. All right, so there's one more question. So it says, thank you for sharing. Last question. What about connection with someone with aunts and uncles on father's side had Alzheimer's, and a cousin, some of the uncle with Alzheimer, had a Parkinson's-like disease? Is that different? So this is a very complicated question, because a lot of neurological disorders, they're kind of interconnected, and especially if you start talking about our aging population and people that are over the age of 60, there's a lot of comorbidities. So it's not rare to find Alzheimer's, Parkinson's, dementia with Lewy bodies. A lot of these are very intertwined, and sometimes people have both or three of these, and that also mixed with diabetes. So there's very fine lines dividing some of these phenotypes, and definitely genetically we see some connections. There's not a lot of connections genetically between Alzheimer's and Parkinson's, but biologically, they're both diseases where there's one protein. In our case, it's synuclein. In their case, it's tau, that accumulates in the brain. There's a specific area of the brain that is affected. So there's a lot of similarities. But yeah, these families where there's a lot of different phenotypes in the same family, sometimes they're very hard, and a lot of the times what's going on is there are 2 or 3 different genes that are involved in this, and some part of the family have a variance in some genes that cause Alzheimer's, and some of them have genes that cause Parkinson's. So yeah, so again, I would just recommend to those families, if you have a lot of family history of neurological disorder, play brain games, do as much as you can, stay healthy, exercise, do things to try to prevent. But in the genetic field, in most of these families, there's not really a single cause. So there's so it doesn't really go from generation to generation. So yeah, so thank you very much everybody for listening. If you have any questions, please let us know. You'll get the slides after the talk. And again, I'm hoping to be there at some point physically, so I can visit you and drink some of the excellent Oregon craft beers and enjoy the Pacific Northwest again. Thank you.
Thank you, Nacho, for sharing your personal connection and also sharing your knowledge. And we also hope to see you in Oregon sooner than later. So that concludes our webinar for today. Just a few things to go over. One, we'd like to thank our sponsors for some educational support that they've provided. Also, there is going to be a video recording of this webinar available on our Parkinson's Resources YouTube page at the end of the week. If you have any further questions, please don't hesitate to email me. If you have any needs or support, please feel free to call Parkinson's Resources. And this concludes our webinar broadcast for today. Thank you so much. And again, thank you, Nacho, for being with us today.
You're very welcome. Bye.